cell life cycle and mitosis. Okay, this will go a little bit slower for you, and I'm going to do some art therapy up here. The cell life cycle. Okay, now one cell could last for 7 minutes. Some cells could last for 70 years. That's 70. Your neurons, your nerve cells, can last for 70 to 80 years, whereas other cells, like your skin cells, only last for a few days. Okay, 70 years. So to make one into two for a nerve cell, it's going to take 70 years to happen. So we'll talk more about that when we get to histology. We have a phase that part of the life cycle, which is most of the time that it spends in. It's called interphase. And this is a time where the cell sits in between its cell divisions. This is most of the time that it sits in here. You're going to have replication of DNA and preparation of dividing into two cells. All the normal ongoing cell functions occur over here. I had a teacher in high school, and he told me that, or told all of us, that interphase is the resting stage of the cell. Well, that's bullcotton, okay? Because it's anything but resting. It's doing all its normal activities, making proteins, fighting infections. It's doing everything during that time. So the cell doesn't ever rest. I mean, it's not dividing. Yes, that's correct. But it's not resting, it's duplicating, it's doing a lot of different things in there, okay? And then we have mitosis, which is going to make one cell into two cells, okay? And those two cells will be identical to each other, okay? We also have a special part of mitosis, the very last ending of it, and it's called cytokinesis. Cytokinesis is actually dividing all the cytoplasm, all the electrolytes and gases that's in there, and divide that into the two cells. So when you look at this, if this is a life cycle of a typical run-of-the-mill cell, you have basically 45, if this is 60 minutes, it doesn't have to be, but I'm just doing it in a essence that you could understand. You're spending about 45 to 50 minutes in interphase, in the green here, okay? And then you have about 10 minutes or so that it goes through mitosis. So most of the time it's in interphase. And interphase can be broken down into three things. We call it G1, S, and G2. Let me just finish. G1, organelle duplication. It's going to duplicate all the organelles in preparation to have two cells at the end to have the equal amount. So it's got to duplicate it. You have DNA duplication in there. Again, two cells are going to have the same type and the same amount of DNA in both. And then G2 is when all the normal activities happen, protein synthesis, fighting infections, all that kind of stuff, okay? So that's what goes on over here. And then mitosis is cell division, and we have 4 phases in there. We have something called prophase, metaphase, anaphase, and telophase, okay? So it's doing this whole thing like that. Question?
How long was mitosis?
It varies for each cell. I can't, it could be anything from like a couple minutes to 70 years.
Okay.
So which one, right? So it's got a big, it depends on which one. And you'll learn more about that when we get into histology.
True.
Okay?
Overall, usually interface, that's longer.
Interface will always be the longest,
Okay.
or longest
So
phase. Okay? So it gives you an idea about this. Okay? All right, so let's talk about mitosis. Okay? Mitosis is cell division, where we're going to, this is interphase, we're going to make the chromosomes, which is what DNA is. Chromosomes, we have to duplicate them. So duplicating the DNA, the constitution, if we're going to make 2 Washington, D.C.s, we're going to have to make 2 constitutions that are identical. One for the original and one for the new one, right? So we have to duplicate that. Then it goes through mitosis, different splitting up, and then we're going to have, this is called the parent cell. You get 2 daughter cells over here. The 2 daughter cells are identical to each other, and the daughter cells are identical to the parent cell. Okay? Questions about that? Now, we could have things go wrong over here, and that could cause cancer, but for the most part, they're going to be identical.
What happens to the main cell after the 2 are removed?
There is no main cell. Think about it. Right? You're going to take
Well,
this.
the parent, what happens to the parent?
it splits into two. Right? You see what's happening here?
But made two.
No, this is going to split into two. Then this can split into two. That can split into two. Yeah, and that's mitosis. So there's no original per se. The other one doesn't die. It splits into two because we have the DNA duplication and all the organelle duplication. Okay, is that clear? Okay. So let's talk about this now. I kind of draw this picture over here. Yeah, let me see. So I'm going to draw this picture. I wouldn't recommend drawing it with me, only because I'm going to do a lot of erasing, and it's going to irk you. Okay, but it's just more my way of doing animation. So you'll see it on the video. All right. So you have this cell. Okay. Now what happens here is this. We're going to have the nucleus. So this is one cell, and we have a nucleus here. Okay? Now, let me just explain to you about what DNA is. DNA could be in two different forms. Okay? It's still DNA. It's like an encyclopedia set. You have 46 volumes in encyclopedias. Do people still read encyclopedias? No,
I'm sure some do.
but do you know what I'm talking about, encyclopedias?
Yeah.
Okay.
Yeah.
Soon, the next generation, you know. So, but anyway, so, because right now I think people don't even know how to read maps. They refer to the GPS, so, anyway, it's another topic. But anyway, you have 46 volumes of an encyclopedia, okay? Now, I can't really go in there and look to see what everything's, all the information's in there, but it's all condensed. Now they're easy, they're really nice that if I want to take 3 volumes, I can put them on my arm over here and I can bring them to the desk. That's no problem. But it's difficult. I'm going to have to now open it up, flip through pages. It's difficult. So what we do is that we could have them condensed in these books, the binding and stuff, or we could take out each volume, rip out each page, and place it onto the floor. And we put another page right next to it. And we could lay it out, really spread out like this, very thin, but if we wanted to float over here and we could look at everything really nicely without even touching anything, and we could have an idea of what's going on. Does that make sense? We're scanning the whole thing. So we can have them thin, uncoiled, like this. And most of the time, that's what it is. We call that chromatin. That's DNA, uncoiled, uncondensed, and most of the time it sits as chromatin. You can't see it. It's really thin, okay? But it's not a good way to divide a cell because you have all these pages all over. So when we're ready to divide the cell, we're going to take all those pages and bind them together into 46 books. Now it's easy to take books off and divide it into one cell into another cell. This is a condensed form, and we call this DNA chromosomes. So DNA could be in the form of chromatin, all right, during interphase, or during mitosis, when it's ready to divide, they're known as chromosomes. Does that make sense? Okay? Now what's going to happen here is that we're getting ready to divide. So we have in here a few things. First off, the cells, the DNA is very loose, and that's chromatin. And there's also going to be centriole pairs. I know we didn't talk about this. We'll learn about it later, but the thing is we have centriole pairs. We have there and another pair here. Okay? These are organelles that I'm going to show you what they're going to do. Okay? And they're on one side of the cell. Now, it's ready to go through mitosis. It's already duplicated its DNA. And it's getting ready to prepare itself for mitosis. It's going to enter the phase that we call prophase. A few things happen here. First off, we need to divide this chromatin. It's difficult because it's so loose. So this chromatin is going to start condensing on itself so that we can actually see it and it's easy to divide. And they are actually going to turn into chromosomes, which look like little X's. What they actually are, that's one chromosome. Okay? Or not one chromosome. That's what we call a chromatid. Okay? We've got a short arm and a long arm. And the centromere is in the center. But when we have a pair of chromatids, that's a chromosome. Okay? So it's really not this. It's really that. It looks like an X, I realize that, but that's one chromatid, that's one chromatid. It doesn't go across. Does that make sense?
Mm-hmm.
Okay? So you'll learn about that in lecture. I'm just trying to get you into the mitosis thing. So they condense and they turn into chromosomes. Same material, just now are chromosomes, and they're duplicated. Okay? This is what happens in prophase. It condenses, turns them into chromosomes. The nuclear membrane also starts disappearing. And the centrioles will go on opposite sides of what was once the nucleus. So one pair will be here, and one pair will be here. And this nuclear membrane will all disappear. Make sense so far? Okay? That's what happens during prophase. You don't have to worry about early or late, just prophase. So what's happening here in our picture, here's your nucleus. It's in chromatin material inside. You have two centrioles over here, two pairs. And what happens here is that the centrioles will start dividing. One will go on one side, one will go the other. The chromatin material becomes chromosomes, okay? And then we're starting to, okay. So now on opposite sides, the nuclear membrane has now disappeared. And the other thing that's going to happen is that these centrioles start developing what we call spindles, or mitotic spindles. Sometimes they're called asters. And what it is that they draw these lines over here, okay? And either one will be like this, okay? And one will be the two, okay? And what they start doing is they pull on each other. It's kind of like a marionette puppet. This is the handle, there's the strings, and you know what a marionette puppet, Pinocchio. I got no strings to hold me, you know, that kind of thing. So you have this thing like this, right? And so this is the centriole. Here's the spindles, or the asters coming down, and Pinocchio over here is the chromosome. So they're being pulled to either side. It's almost like a tug of war. And then this is going to eventually, when the equal force on either side gets pulled, eventually these green things are now going to line up in the center. So these green chromosomes are now going to be lined up in the center because of them being pulled equally on either side. Think of it like an imaginary disc that will be there, and it's riding along that disc, all right, or plate. When they're all lined up like this, we call that metaphase. Okay. Now the duplicate chromosomes are going to start pulling away from each other, and that's called anaphase, and they kind of look like so their arms are going to go like, it's like if someone's pulling me, like a tugboat is trying to be pulling me, my arms kind of go like that. Oh, it doesn't look so good, but it does that thing, okay? So what is happening is that they're being pulled and the arms are like going backwards. I'm creating these V's going like that. Okay? Then what's going to happen is the whole cell is going to start dividing. And now this has the original chromosomes before it was duplicated, and you're going to start having the new nucleus forming here and a new nucleus forming over here. But there's going to be something in the middle here during telophase. We call it a furrow. It is not divided all the way, but you're going to see a nice furrow where it's just, and you'll see what I mean by the picture. It's going to be like where it's going to divide. Okay? This is also the time when cytokinesis also occurs, the division of the cytoplasm. So here's where metaphase is. They're all lined up in the center there. Anaphase, they're being pulled away from each other. And here's telophase, where they're starting to, and also the chromosomes will uncondense, and it'll turn, the asters will disappear, the centrioles will go on the outside, and the chromosomes will turn back into chromatin. Until it's now two separate cells, and now that's two separate cells. It's not telophase anymore. If it has this furrow, it's telophase. So here's interphase, and you're going to see a lot of interphase in the cells because that's most of the time it sits in. You can start seeing the condensing of the chromosomes. That's the prophase. You could have them all lined up together. That's going to be metaphase. They're going to be pulled apart. That's anaphase. Here's telophase. See that wonderful furrow in between? Okay? And that's telophase. So you can use that mnemonic ITMAT. It's not my mnemonic. It's, yeah.
Is the cell always in one of those stages? Like,
Yep, it's usually in that stage.
Okay.
But that's where they come in, like, you know, is it late prophase or early metaphase? You know, it's going to be something like that. I'm not going to give you something like that. I'm going to say, I'm going to give you something like this.
So they're like always replicating themselves.
Not always replicate themselves. It depends, like something that's going to take 70 years, it's not going to replicate.
But it's in the process.
No, it's an
somewhere.
interphase.
But that's part of the process.
No, that's not the process of mitosis because they're not duplicating themselves. They're doing the normal stuff like fighting infections, they're making proteins, it's doing other stuff.
This soccer player's knee will be as good as new in a few weeks, thanks to mitosis, a type of cell division that generates new cells for growth and repair. Let's move into a cell to witness the events of mitosis. Before a cell can divide, it must first duplicate the chromosomes stored in its nucleus. During chromosome duplication, several bubbles open up along the chromosome. Each bubble grows until it merges with an adjacent bubble. Each chromosome now consists of two identical copies called sister chromatids. Getting closer, we see that each sister chromatid consists of DNA wound around small proteins called histones. The sister chromatids begin to coil into tight helical fibers. Outside the nucleus, centrosomes that duplicated earlier move away from each other to opposite sides of the cell. Microtubules extend from the centrosomes, forming the mitotic spindle. Back in the nucleus, the DNA forms loops, becoming more compacted. These structures fold back on themselves, eventually condensing into a shorter and thicker chromosome consisting of two sister chromatids. As the chromosomes continue to condense, the nuclear envelope breaks up. The array of spindle microtubules is now extensive, and the chromosomes are fully condensed. Spindle fibers from each pole attach to protein structures located at the centromere of each sister chromatid. As the chromosomes are bound by spindle fibers from opposite poles, they move first one way and then another. The counteracting forces of the spindle eventually cause all the chromosomes to end up at the center of the cell, as if arranged on an imaginary plate. The sister chromatids are released from each other, each becoming a full-fledged chromosome. They are moved toward opposite poles of the cell, pulled along the spindle fibers attached to them. At the same time, overlapping spindle fibers that are not attached to chromosomes continue to lengthen, pushing the poles farther apart. Once the chromosomes arrive at their destination, they become less condensed. Two new nuclear envelopes form, completing mitosis, the division of one nucleus into two genetically identical daughter nuclei. The cytoplasm divides by the process of cytokinesis, forming two separate daughter cells. In your body, millions of cells divide every second, providing new cells for growth and for repair of damaged cells.